evening, everyone, and welcome to this very special event brought to you by Charnwood Forest Geopark. My name's Jack Matthews, and I'm going to be the chair this evening. I'm the Geoheritage Officer at Charnwood Forest Geopark, and I want to start by saying a big thank you to the National Lottery Heritage Fund, who financially support a great deal of the work we do. This is a special event also because it's British Science Week, and it's the very first of these online public talks that we're hoping to continue to bring you over the year and into the future from Charnwood Forest Geopark, celebrating not only the geological significance of Charnwood Forest, but all the many things that make our region so special. Before we get on to our two fantastic speakers tonight, two for the price of one this week, just firstly, an apology to those of you who are trying to join us on Facebook. We've had a bit of a struggle getting the connection going there, but the platform is working fine for YouTube and Twitter. So if you've got a friend who's trying to make it into the event, they can still use the YouTube or Twitter link and they will see all of the event. I can also say that our speakers have agreed to the recording being made public. So we'll make all of this available via our various outlets as well. And the other thing to say is our speakers are going to give their wonderful presentation about the secrets of Groby Pool mud. If you have a question at any point, please do let us know. We're going to have a Q&A session at the end of the talk, but it really helps me if you let me know your questions as you think of them, so you can pop them in the comments and the chat if you have an account on the platform that you're looking at, or you can message me via text or SMS and I will pop up periodically through the talk my details, so you can see there. Pop your questions as you think of them, and you'll see this banner popping up, and I will put it in the chats also. You can text your questions to me at 07980 794 827. But at that point, I think it's best we bring our wonderful speakers into the screen today. So we're joined by Professor Helen Bennion. She is Professor of Environmental Change at the Department of Geography at University College London. She's been there since 1985, and her research and teaching focuses on aquatic ecology and palaeoecology as well. And we're also joined by Professor Carl Sayer, who is also at the Department of Geography at UCL, and he leads the Pond Restoration Research Group. Welcome to the both of you. Thank you so much for joining us.
Good evening.
Thank you.
Fantastic. So I think at that point, we're really pleased to have you here to talk about the secrets of Groby Pool mud, a place that many of our viewers will know very well indeed. And I think we're going to all learn something new about this local landmark. So I'm going to pop the slides there. I'm going to disappear, and Carl's going to disappear for the moment because Helen's going to kick us off.
Okay, well, thank you very much for the kind introduction, Jack, and for the opportunity to give this talk this evening. It's very much a double act, as Jack said. I'm Helen, obviously, and you'll hear from Carl later. So I'm going to kick off and I shall return later, and then Carl will provide the exciting meaty sandwich in the middle with all the exciting stuff. But we thought, having called this talk The Secrets of the Groby Pool Mud, we should probably start by saying a little bit about why we're excited by mud. You can see us both holding up our various muddy trophies there in the slide. But we'll introduce you very gently, I think, initially to our research world of palaeolimnology, which perhaps doesn't trip off the tongue that easily, but it's literally paleo as in past, and limnology, which means lakes. So it's the study of past conditions in lake systems from evidence in lake muds, which we refer to as the lake sediments. And I've tried to capture in this slide some of the things, the sort of information that we can gather from sediment records. So you can see on the left there, I've identified past conditions next to my thumb. So that's obviously the mud that's been at the bottom of the core, which was deposited in the furthest back in time. And then as you sort of move up through the record, it's really like opening the pages of a book, sort of revealing the story and the history of that lake. So as you move up towards the top of the core, which you can see is identified as current conditions, that's the sediment that's just been deposited in the very recent past, just the last few years. We can obviously track all sorts of changes through the time period represented by that record. So that might be, as I've indicated there, chemical change, could actually be physical change. But what we're going to concentrate on in the presentation tonight actually is more the ecological information, the ecological change that we can glean from these sediment records. And then on the right of the slide, it's the sort of the so what, so what, that's all very exciting and very interesting, but what does that actually mean and what's the relevance in terms of management? And so much of the information that we can achieve through analysis of sediment records can tell us an awful lot about pre-impact conditions, which we often refer to as baselines or reference conditions. That's the sort of terminology that's used in things like if those of you might be familiar with the EU Water Framework Directive, where you kind of understand the ecological status of a site by comparing what it's like today with what it was like in the past in these when it was at these sort of baselines or reference conditions. And then, of course, we can look at how our sites have responded to a range of pressures. So they might be multiple stressors. And I think really importantly for management, when there's been some kind of management intervention or change in land use in the catchment or land management, we can also use these records to track whether the sites, the lakes are starting to recover. What's the extent of recovery? Are they recovering at all? And of course, that then helps to inform restoration goals and setting targets for future management projects. So we can gather a lot of information is the takeaway. So how do we do it? Well, here's a few slides of sediment coring. We've got lots of different tools to collect sediments from lakes. I've just got a couple of examples here. So the one on the left is what we call the Big Ben corer, for no other reason that it's big, and it was co-designed by our colleague Ben, so it became known as the Big Ben corer. And this is what we call a piston corer. So it's very good for taking long tubes of sediment in shallow waters, and you can use you can see the rods there that my colleagues are holding. I've got the glamorous job of lying down. That's me pushing the bung in at the bottom of the tube, and you can maybe see the white circles at the top. That's a sort of a piston that slides up as we push down. So it provides these really nice long intact records from shallow systems, and it's just, you see it's being used just from a sort of little platform attached to a couple of inflatable boats. And then on the right, in contrast, that's what we call a gravity corer. So they're really useful for deeper water, where we can just lower it under its own weight on a rope. Generally for getting shorter sequences, you obviously, are limited by the length of the tube that you can use here. And that triggers either with a messenger or under the sort of slack, when the slack goes off the rope under its own weight. So we use those in deeper systems. So we have lots of tools at our disposal for taking different types of cores, depending on the nature of the site. And the one I'm showing in this slide is what we call the Livingston piston corer. And this is actually the gear that we use for collecting most of our cores from Groby Pool, which we shall obviously move on to later. So again, very similar to the Big Ben corer, it's just slightly narrower, and it allows us to push it into the mud and get these nice long records of up to 1 or 2 meters long. And then, of course, we have to what we call extrude the cores, or sort of sample them. This is a typical little extrusion rig, and it's really just a way of winding up the sediment and slicing it slice at a time. And you can do that at known intervals, selected intervals, depending on the accumulation rate of the sediments in your particular site, or the kind of questions you're asking of the sediment. So typically a few millimeters to a centimeter thick, we would slice them, and then each of those slices can then be analysed for a whole range of parameters. And I thought it was a good opportunity perhaps just to put up this little cartoon, which just very simply explains what the sediment record can capture. And we're not obviously going to talk about all of these this evening, but just to really make the point that it's the sediment core is there at the bottom of the lake, as you can see there in that sort of deepest point in the deep basin. And it can actually capture catchment inputs. So, you might be familiar, for example, with pollen remains from vegetation in the catchment. It can capture the atmospheric inputs, particularly atmospheric pollutants and toxic metals, for example. But again, for the purposes of today, what we're going to really focus on are the in-lake inputs that are captured in the sediment record, and particularly the biological remains. So all the different biota, like the algae, the aquatic plants, the animals living in the lake. And obviously, as they die, the organic matter rains down and collects and forms our sediment core and hence our record. So obviously, when we have our tube of mud, we know how deep that is, but now we have the challenge of converting that depth into a known time period so that we know what time period we're actually dealing with. And that's where we come into core chronologies, and we have a whole range of dating methods at our disposal that we can use. And I've just shown on here the two that we use most commonly for looking at the last 150, 200 years, which is, again, the sort of time frame that we're going to talk about this evening. So some of you may be familiar with these, actually, but the common one is lead-210, and that's a naturally occurring radionuclide, which has this half-life of 22 years. So it's as it declines in the sediment core and the concentrations, you can then work out the dating for the last 150 years or so. And then we can also couple that with cesium-137, and that's actually produced by nuclear fission, and it's produced obviously by the nuclear process. That has a similar half-life to lead-210. And so atomic weapons testing in the 1950s and 60s leaves markers in the sediment record. And then most notably, if you, I'm sure you'll all remember the Chernobyl disaster at the plant in 1986. And that generated this enormous cloud of cesium, which you can see on the globe picture there, shown by the red patterns, which dispersed atmospherically across huge amounts of the Northern Hemisphere, particularly across Northern Europe. And then obviously it found its way into the soils and ultimately into our lakes. So we have this very strong 1986 marker as revealed through the cesium activity from the Chernobyl accident. And that's just shown here by a core that's taken from a Scottish loch, which has these really very clear peaks. You can see on that little graph there, I've identified showing the 1963 atomic weapons testing, and then another peak which corresponds to Chernobyl. And just by way of sort of contrast between, we work on obviously lots and lots of different lake types, and Groby Pool is obviously a lowland catchment, quite a productive system. But just by contrast, upland lakes would typically have 1 or 2 millimeters per year of sediment accumulating. So one of these short 40 centimeter cores could represent several hundred years. Whereas a site like Groby, they would have an accumulation rate of about 5 to 10 millimeters per year, so maybe up to a centimeter a year. So again, one of these 40 centimeter cores could represent less than 100 years. And we actually do have dating records for Groby Pool. So here are the dating results, and you can see the cesium peaks in that top right graph, again helping us to constrain where these key dates are. And then we have this little age depth graph in the bottom. And that's really just showing you for one of our cores, which was collected in the year 2000, the year 1940 lies at about 35 centimeters. So we're getting about 60 years of sediment in that sort of 30 to 40 centimeter core record. So some of the cores we're going to show you later are much longer than that. They're about 1 meter long. So, we're getting a good couple of 100-year record from that length of sediment. We also have another tool that we can use to constrain the chronologies of our sediment records. And this is these are the spheroidal carbonaceous particles, which we call the SCPs. And as you can see in the little photograph there, these are the particles that are produced from combustion of oil and coal. So they're actually emitted from power stations. And because there's a very clear industrial record of the burning of fossil fuels, we can use this very classic standard profile, which is shown in the graph here on the left, to constrain these key points in history in our sediments. So this is actually a record from Groby Pool. This is a core we've called Groby 4. You'll realise as as the talk progresses. We've taken a lot of sediment cores from Groby Pool over the years. So yes, you can see the three key markers. You've got the first presence of these particles around about the mid to late 19th century. Then you get a rapid increase in the concentration starting in the 1950s as we were burning more fossil fuels. And then these peak around about the 1960s and 1990s. That depends from one record to another. But obviously then after the energy crisis, the concentrations start to decline. So these, again, another really good way of getting a good handle on the dating of our records. So moving on then, if we think about the timescales that we're interested in, this is just a nice little graphic that summarizes the time frame of many of the human impacts that are exerted on our lake ecosystems. So obviously we've got a lot of long-term impacts. You can see there sort of over 10,000 years, things like long-term climate variability and lake catchment processes. But for the purposes of much of our research really is focused on that much more recent past. So really post 1800 AD. And you can see there by the little sort of fatter red bars just how many different pressures there are on our lake ecosystems over that more recent 200-year timescale. And I've highlighted in green there really the ones I think that pertain most strongly to Groby Pool, and will form the focus of our presentation when we kind of get into the Groby detail later, but very much around nutrient loading and the issue of eutrophication, and to some extent sort of habitat disturbance that perhaps comes with that. But you can see there's a whole host of other stresses that impact on systems, often in combination. And I think that the beauty of the sediment record is obviously we've highlighted there the present day, and of course monitoring, current day monitoring gives us that understanding of the ecological condition today, but gives us no understanding of really how that's been reached. And of course what paleo does is allows us to actually track where these lakes have come from. Because they're nutrient rich today, have they always been nutrient rich? Were they more, nutrient poor in the past? And that really gives us then a much better steer as to what the chances are of restoring some of these sites in the future. What are the ongoing pressures? What are the continuing threats? And hence, what can we set meaningfully as feasible targets for recovery? And sadly, and I think many people watching this will be aware of this fact, that freshwaters are thought now to be one of the most endangered ecosystems in the world. If you've seen the recent Living Planet report, you'll see that both the magnitude and the rate of species loss in freshwaters is now exceeding that in terrestrial systems and in marine systems. So, it's a real wake-up call, and there's lots of urgent questions that we need to be addressing. And as I mentioned in the earlier slide, nutrient loading is of particular interest to us, and particularly relating to lowland catchments such as those of Groby Pool. And this is a review that came out recently by Penny Johns and colleagues, and it's just a really striking graphic here of showing the global distribution of the nutrient loads of, this is phosphorus, to fresh water in recent decades, showing that losses from land to fresh water have almost doubled in the 20th century, and actually quite similar striking statistics for nitrogen as well. So we're really pumping a lot of excess nutrient into our systems. If we just sort of drill down and look at some of the key sources of those nutrients, this little cartoon obviously captures a catchment where it's being hammered by absolutely everything. I don't think that ever happens in the real world, but obviously just to sort of highlight some of those key sources in terms of sewage effluent and industrial effluent, household waste, and then the more diffuse sources such as intensive agriculture and aquaculture. And you can imagine, I mean, we've been aware of eutrophication since the 1960s. And so some of these catchments have been subject to these high nutrient loads for, best part of 40, 50, 60 years. And in shallow lakes in particular, that's generating this issue of internal loading. So you've basically got this inbuilt reservoir, this inbuilt store of phosphorus that's built up over those preceding decades. And under certain conditions, that phosphorus can then be released back into the water column. So, even when you've reduced your external loading, you could still have these ongoing internal loading problems. And that particularly happens when sediments are deoxygenated, when you've got bacterial activity, And also, as you can see in the little picture there, through sediment resuspension, through particularly like the activity of benthic fish such as carp, that's sort of foraging amongst the sediment, and that obviously allows the phosphorus to be released back into the water. So it's not a problem. It's a problem we quite well understand, but certainly hasn't gone away despite being aware of it for, you know, the best part of 60, 70 years. And again, I think we know only too well some of the consequences of eutrophication. That increase in lake productivity brings with it increase in algal biomass, and typically these blooms of microscopic algae, which have often been referred to as harmful algal blooms. Most famously, perhaps in the Chinese Olympics, when some of those lakes that we used for the games were just covered in this sort of paint slicks of cyanobacteria, these blue-green algae. But not only that, you can get dense mats of floating macrophytes, and ultimately the loss of the submerged plants, which play a really key structuring role in lakes. So the loss of those has really profound implications for the rest of the system. And then with that, we get oxygen depletion, deterioration in water taste and odor, which is obviously a big concern for the water industry. Clearly a reduction in the aesthetic quality, and a problem really for delivering on a whole host of ecosystem services that freshwaters provide. So obviously biodiversity, conservation, amenity, water supply, fisheries and public health. So, you know, the need to tackle eutrophication, you know, is really a great need. And sadly, shallow lakes, and, you know, Groby Pool obviously is a shallow lake, really have been sort of felt the sort of most profound impacts of eutrophication. And what we know both from our paleo work, but also long-term records and observational data, is that many of our shallow lakes have sadly shifted from these wonderful, diverse, clear water, plant-dominated lakes that you can see in the top photographs there, which have lots of refuges amongst those plants to support invertebrates and fish. So they have balanced fish communities, low algal biomass, and you can see the beautiful clarity, you know, the clarity of the water there, and these wonderful underwater carpets of plants. But sadly, many of them have shifted to these rather horrible, turbid, plankton-dominated green soups that you can see in the bottom part of the slide there. So often impoverished plant communities, or indeed in some cases complete loss of the plants, as you can see in some of those slides. And that has a really poor light climate, so nothing can grow under these algal mats. The sediment phosphorus release that I mentioned earlier, which becomes even more problematic under these conditions, deoxygenation, and then ultimately, of course, fish kills in many of these sites. So where does our research come in against a backdrop of eutrophication? And hopefully now you've kind of got an understanding of how we can use paleoecology. So I think really our main interest in recent years has been trying to understand changes in lake structure, so how they've changed ecologically, and then trying to get a handle on how the function of lakes has changed as well, and to do that over these timescales of decades to centuries. So some of the key questions that our research has been addressing have been, how have lakes responded to enrichment over these timescales? What were the former nutrient concentrations and the associated plant and animal communities? And then have lakes recovered when there's been some sort of management intervention? And if not, why not? You know, what factors might be confounding lake recovery? And actually climate change has been a big sort of angle on that in recent years. So we'll move on now to give you a few more sort of insights as to how we actually get at some of this ecological information. And this is my last slide before I hand over to Carl to sort of fill in some of the details and to take you into the wonderful world of Groby Pool. But I think it's fair to say that Carl and I's first love was diatoms. And that's a picture of me looking very young doing my PhD in the early 90s. And Carl did his PhD on the diatoms of Groby Pool, which you'll hear more of later. So for those of you who are new to diatoms, it's an absolutely wonderful world, and we could dedicate a whole talk just to that alone. But so just very briefly, they're the siliceous algae. They're single-celled algae with these silica cell walls, which have this wonderful sort of intricate patterning, which allows us to identify them to species level. And they're resistant to decay, which is why they're so great in sediment records, because they preserve so well. And really importantly, they have very clear ecological requirements for a whole range of water quality parameters and have associated habitats. So when the circular ones you can see there are typically what we call the centric diatoms, which live free floating as plankton in the lakes. And then some of those which are more the sort of little oval forms, that what we call the pennate forms, they're attached diatoms, which might be attached to the mud surfaces or sand grains or rocks and stones, or indeed to plants. So the epiphytic diatoms that attach themselves to the plants. So we can glean a huge amount of information by looking at diatoms in sediment records. But Carl will now explain to you all the other wonderful remains that we can find in sediments, because whilst I'll say our first love was diatoms, we have learnt that diatoms alone don't tell the whole story. So over to you, Carl.
Thank you, Helen. Next slide, please. Lovely, thank you. So the diatoms are some of the most beautiful, ornate things, really, and they tell us so much. And you had my favourite diatom on there, Helen, Epithemia sorex. Sure the audience all recognize that. But anyway, there's a whole host of other things that you find in sediment cores. And in fact, there's probably more things you can find in a sediment core than you can in weeks of surveying the lake for contemporary organisms. So many things leave a trace in the sediment. So the Cladothera, the zooplankton leave eggs and parts of their body. All sorts of invertebrates, such as chironomids, leave their head capsules and different parts of their bodies. Plants, both around the lake and in the lake, leave remains, seeds, spines, all sorts of little bits that we can identify. And even fish leave their scales and bones. So there's very, very few things that don't leave some kind of remain in the sediment. And we can use that not only to understand how things have changed sort of chemically, but also how the whole biological system has changed, how the food web has changed, how things interacting with each other have changed. So it's a fantastically powerful approach. Next slide, Helen. And of course we can use pollen as well. This has been done at Groby Pool, so we'll cover that a little bit. Carol David did a wonderful PhD on Groby Pool. It's one of the first bits of work. But anyway, we can use pollen grains to reconstruct the vegetation around the lake. And that's been particularly brilliant at looking at long-term evolution of the vegetation in Britain, in response to sort of climatic changes, and also more recent changes in vegetation in catchments. Next slide. So let's go to Groby Pool. I started working on Groby Pool because I was at Loughborough University for my PhD, and we were looking for something for me to do. And I kind of discovered this lake, and I went to visit, and I thought it was fantastic. I had no idea about its history, but because I was at Loughborough and Groby Pool is right next to Leicester, it seemed like a good place to go, really. And it was the biggest bit of luck ever, really, because it's the most fascinating place. So it's a shallow lake. Its average depth is 1.1 meters. It doesn't get any more than 1.8 meters. It's obviously lowland. Has a small, mainly rural catchment. We'll look at that a bit more in a minute, but pasture, arable, and some old woodland in the catchment. The waters are relatively alkaline. The pH is relatively high. And of crucial importance, really, to this talk is that it's very eutrophic today. It wasn't anywhere like it is today in the past, but the current phosphorus values are, when you look at an average there of 680 micrograms per litre, that's really high, okay? And as a consequence, we've got lots of algae. So chlorophyll A there, average of 10 micrograms per litre, that's really high. That means quite green water. But anyway, there's Groby Pool. Let's start off and try and understand it, and also learn about its history. Next slide. Thank you, Helen. The age and origins of Groby Pool have been the subject of much debate. Traditionally, and I'd say up until the 1990s, you could read lots of books and local literature which describe Groby Pool as the largest natural sheet of water in Leicestershire. So it was regarded as a natural lake. The earliest reference comes from the 16th century, where Leland refers to Groby Pool as a fair and larger pole as likely is in Leicestershire. So it's referred to quite early on, so it's clearly very old. But sediment coring in historical studies by Carol David, who was the PhD student preceding me at Loughborough, determined really clearly that it was an artificial lake. The sediments just aren't deep enough for a natural lake. And there's also quite a lot of evidence that it is human-made. So it can be traced, if you look at the name of Groby, named after the village, the Danish words grob and grop mean pit or hollow. And that, when you hear the word pit, it suggests digging. So this clearly suggests it was a flooded working of some kind. There's no reference to the site in the Domesday Book, which makes you think it wasn't around then. But an Inquisition post-mortem of 1288, conducted on the estate of William de Ferrers, refers to 2 fish ponds and 2 associated watermills. And one of these, we think, was surely Groby Pool. So it looks like it was sometime in the, 1200s or something, maybe a bit before that, Groby Pool came into existence. Next slide, Helen. Just to show you how this kind of debate over its origin has continued, really. I was just researching a little bit more about the lake because we haven't worked on it for a long time. So I was wondering what else has been going on. I saw a living landscape report by the Wildlife Trust in 2009, and they described it as an ancient piece of water, which is using some of the old language, really, and that and it's described as the largest natural water body in Leicestershire. So it's referring to some of the old stuff. And then it says, John Martin, former keeper of geology within Leicester Museum Service, believed that the present lake is 19th century artificial water body. So there's quite a lot of debate, really, but I think really, from Carol David's work, we can sort of place it in the sort of early medieval era. So, okay, next one. So what do we know about it? Well, Carol David did some wonderful work putting together historical records of land use in the catchment. And we can see from this, the catchment reconstruction, this is from wonderful mapping in 1757. So in that year or that era, it was 25% old woodland and the remainder was pastures. So there was no arable land in the catchment. But as we move into the current era after 1900s, 1925, 30% of that pastured land had become arable. So there's a lot more arable land in the catchment, and arable land means losses of soil and nutrients to streams, which you can see there feeding into Groby Pool. So you can guarantee really that this major changes in the catchment, had an influence on Groby Pool. Next one. And here shows the catchment today, really, or part of the catchment today. So you can see what it's like, really. There's a lot of rural places, but we've got roads, we've got quarries, we've got arable land, and with all of those things, we're going to end up with impacts on Groby Pool because the lake really reflects very strongly what's going on in the catchment. Okay, this is a diagram I got from some unpublished, wonderful thesis work of Jane Beresford from 1989. And she shows these maps of the Groby Pool catchment. And you can see in 1700, there's just Groby Pool. There's no other standing water in the catchment. By 1800, you can see Groby Pool's got lots and lots of ponds keeping it company. There are lots of dots on the map there. By 1900, in the bottom left, you can see we've now got lots of lakes, there's lots of reservoirs. And then by 1979, we can see actually we're starting to lose water bodies as ponds are being filled in and things like that. But the key thing really is what it was like in the early days in 1700 and after, is that there was no other major lakes in the area. And that made it a mecca for people to go and visit. We didn't really realize this. But so many people who are interested in botany, so that might be reverence and perhaps more wealthy classes, spending their time recording plants, went to this lake because it was, it was a place they could record aquatic plants. And also it had the most remarkable flora in the early days. And so all of these plant records are incredibly useful to us. And Jane Beresford, and I added a bit more to it, collated the wetland plant data back as far as 1747. So the first plant records come from 1747. There's also some really good, more recent work by Edith Hethelgraves and other people in the 70s, 80s, 90s. So lots and lots of plant records that can tell us a lot about how this lake has changed, and we can compare that with the sediment record, as you'll see later. Okay, so just to show you some of this lovely information you get from historical readings and records and notebooks and things like that. This comes from the Leicester Literary and Philosophical Society, 1899. I'm sure you've all read this. It was after much effort that a party was got together. So they obviously struggled recruiting in this organisation, unlike you lot. But we started 11 strong, proceeding to Groby Pool. There we joined Mr. Turner. Only the margin of the pool open to the road was searched, so a bit lazy there. And I think anyone's been to Groby Pool will know that little bit of ground. But there was a good display of ranunculus flammula from which Mr. Bell picked specimens with 6 petals. So we've got a record of ranunculus flammula there, which is quite a scarce plant, really, but it's recorded for the lake. Anyone tell me any details on this picture? I'd love to see it. It's an old photograph I found. Not learning a lot about the plants there, but it's wonderful to see this lake and people interacting with the lake in the early days. I'm guessing that's late 1800s, early 1900s. But anyway, as you come through to the modern era, you start picking up a few old photographs, and they then start to tell us about botany. So, and we can add those as records to the database. So here we've got a picture of Groby Pool in 1955, and the plant that we can see, and it looks really clear there actually, just from my sort of trained eye and looking at a lake and what the surface looks like. It looks like really clear water in the distance. But we've got big beds of Nymphoides peltata. That's the fringed water lilies in that picture in the left-hand corner there. So we've got records of that from 1955. Next one, Helen. In the late 1980s, there's a picture from Carol David. The Nymphoides had gone by this point, disappeared, but we had remaining large beds of Nuphar lutea, which is the yellow water lily. And that's a real symptom of eutrophication, to just have these sorts of things left, because they're water lilies, they're putting the leaves up toward the surface to get the light. They can cope well when the water's starting to get a bit more murky. Next one. And so here we've got a reconstruction that comes from Beresford's work, from Jane Beresford's studies, looking at the changing aquatic flora of Groby Pool. So these are wetland plants. So, and before 1850, this is, even though, you know, we've got far less biological records at this point, there was still 75 species recorded for Groby Pool, which is absolutely remarkable for a lake. And 36% of those are submerged and floating leaf species. So believe me, that you know, from knowing lakes, it was an astonishing lake in this early period. And what we see over time really is the number of species sort of declining. Particularly look at the right-hand column, you see the number of submerged and floating species declining. So even though recording is increasing, and we've got many more people visiting the lake and making doing surveys, and you know, full formal surveys, the number of species are recording. So this is very clearly showing a decline in the plants, which reflects a decline in Groby Pool. Next one. What are the early species losses? Well, we look at before 1850 and 1900, there were some amazing species present. So Utricularia vulgaris, Littorella uniflora, the shoreweed, different Potamogeton species. And there's an amazing plant called Damasonium elisma, the star fruit, as well in the top right picture there, which was present pre-1900. That's an extremely rare species I'll come on to in a minute. And 45 species have been lost from the site since 1747, when the first records were made. Next one, please. And this just shows the historical records of some of these plants put together. And the key thing really is on the left-hand side. You can see the black bars there, and there's an arrow showing shoreweed. We've got a number of things on that left-hand side which are disappearing, you know, before 1850, present early on, which are really the hallmarks of what we call a mesotrophic or even slightly oligotrophic lake. So very low nutrients, very clear water, and you have a very complex structure amongst the plants, in particular very much more sort of prevalence of low-growing species such as the shoreweed. You can see there that don't have to grow high because there's plenty of light coming down to the bottom of the lake. And then if we move through time, we'll see if you go from left to right, the species that occur more recently are much more associated with more eutrophic, low sort of light penetration, a bit more murky conditions. So we see a very, very clear transition from eutrophic, high diversity, complex structured flora to this much more simple eutrophic flora over 250 years or so. Next one. Just to highlight the importance of the site really and the past in particular. So here's Littorella uniflora, the shoreweed, a big red dot there. That would have been really common across lowland England into eastern England hundreds of years ago. And then more recently, you can see it's disappeared from most of central eastern England, even large parts of southwest England. It's only found in sort of northern Britain and more sort of upland places, really. And Groby Pool is now extremely isolated from this plant. You can see there's a few modern records around it, but not many. So not a great deal of chance of this plant recolonizing unless we're very lucky. But just shows you, the importance of this lake in the past in having species which are, quite rare today. In the Midlands, really, it was a very, very important site in the Midlands of having, scarce plants. Next one. Bladderwort is another thing that it had before 1850, and you have to imagine really, and I've seen it a few times in my life, a lake covered in yellow flowers, just how beautiful that might be, and that's what Groby Pool was surely like. Bladderwort's an incredible plant really because it's a carnivore, so it has a little hair trigger on it, and it pumps out the water and creates a vacuum. And then a little tiny crustacean comes on, like a daphnia that you can see on the right-hand side, touches the hair trigger, opens the bladder, and then the bladder swallows up the invertebrate and digests it. And that's really a very good adaptation to low nutrient conditions. If you can't get it from the water, you get it from animals. And so that's why this plant is, very much associated with low nutrient conditions, but yeah, not present anymore. Next one. And here's one to look for, and I said to dream of. This is Damascenia melisma, the star fruit. So it was used to be found in Groby Pool. It was lost sometime before 1900. But anyway, you can see there it was there, the red dot, and it had a few other sites around it in that era. It's now only found in a very few sites in southern Britain. So if anyone sees that in Groby Pool, please call me. It was one of the most special plants that the lake had. Next one. So let's get onto the palaeoecology of Groby Pool. So we've got very, very good evidence there of changes in the site. Its catchment has changed. We can see that from historical recording, actual records, the lake has changed. But the palaeoecological record, because it's continuous, can tell us so much more detail, and that's why we've used it. So our questions really were, how has the lake changed over the last few centuries? And why has the lake changed? And just to sort of add to the sort of bigging up Groby Pool, it's probably one of the best studied lakes in the world, really, for for knowing its eutrophication history and its story and how eutrophication has affected all these different layers in the food web. There's very few places like it. And so it's surprising there's any mud left in it, really. We've taken lots of cores from it since lots of people have been coring it since the 1990s, and this is the sort of picture that we have at the end, comes from all these cores. So next slide. So here's the first thing I want to show you. This is the diatom algae, and so we can see on the left-hand side we've got depth, and you can see some dates on the right-hand side there. So at 60 centimeters, you can see you're at 1845. Anyway, the key thing to get from this, without giving you all sorts of detail about the diatoms, is a very clear eutrophication story. In the early days, before 1845, we've got a real absence of the plankton. The plankton just isn't there. The diatoms are living on the bottom, attached to plants, attached to the mud. there's not enough nutrients to create a plankton. So the water is, is like gin, it's crystal clear. After 1845, and there's probably reflecting changes in the catchment, we're seeing a little bit of a rise in the plankton there, and that sort of continues. And that's those species on the left-hand side. There's first 3 species really there. And then in 1937, when the Slate Brook was fed by sewage from the sewage works on feeding into that brook, that affected the lake. And we can see that we got very clear changes at that point, and a big increase in Stephanodiscus parvus, which is the diatom in the top right corner there, which is a real hallmark of eutrophication. And that's telling us we've got quite murky water in the spring and probably in the autumn. So very clear evidence of eutrophication and the stimulation of the plankton. Next one, Helen. And then we've got macrofossils. So these are remains of aquatic plants that change through time. And you can see there's a real succession there as you move from the left-hand side to the right-hand side. Before 1800, we've got lots of things called stoneworts in the lake. So these are plants which form dense meadows on the bottom of a lake. They clear the water in themselves, and they're associated with clear water. And it provides an extremely complex and interesting place if you're an invertebrate to live in. So you can expect very, very high diversity, at least before the 1800s there. But as you might expect from the diatoms, telling us more plankton, the stoneworts disappeared after around 1800, and then we've got a succession going left to right to more and more eutrophic species on the right-hand side there. And that's also associated with a large decrease in diversity if you look across the macrofossil record. So macrofossils of aquatic plants tell us very clearly that the lake is becoming eutrophic, and it's losing plants, it's losing diversity, and it's losing its complex structure. Next one, Helen. And then I think this is my last one. This just shows you really a reconstruction of what Groby Pool was like underwater before 1800. So this is what you would have expected if you could have dived down, seeing these dense stonewort meadows, and then arranging amongst that and on the edges and up into the wetland and other places, a whole host of other scarce species. It really would have been one of the most astonishing places. And we have a really, really good knowledge of what it was like. So is that Helen? Yeah.
Okay, thanks, Carl. So yeah, Carl's obviously walked you through the fascinating botanical history of the site. So in this last section, what we thought we'd try to do is really now try to sort of bring it all together and sort of paint this more coherent picture of what we think's been happening at Groby Pool, and not just what's been happening, but why. So so this is what I've always called, I don't know why I call this the orange plot, but we showcase this a lot actually. As Carl said, Groby Pool is really this kind of archetypal system that really showcases the sort of dynamic sequence of changes that you see in shallow lakes associated with eutrophication. So that summarizes the plant macrofossil data and the diatom data that Carl's just shown you, but also brings into the picture some of the other remain types that you saw in that early indicator slide. So we've also got notably shifts in the zooplankton, so much more of these large Daphnia, the water fleas, and we've got shifts in the sort of habitat association of the chironomids, so the midges, the non-biting midges that are in the system as well. But you can really see there with the line drawn at 1800, 1850, just how different the system was prior to that time, and this sort of dynamic changes that have happened since then. So what we want to do in this sort of last section is try and sort of really get into the detail of this a little bit more and try to explain why we think we're seeing these changes. So just a quick recap, really. What we tried to do in this, we published this work in a paper, golly, I'm surprised to see that's nearly 10 years ago now, but a paper in freshwater biology. And as Carl said, we've taken so many cores from Groby over the years, and there's quite a lot of noise if you look at every individual record. So what we wanted to do in this paper was to try to combine all of the evidence that we had, and to remove some of that noise by looking at the changes over these little 20-year time slices. So that's what all those bars represent. So each one of those represents a 20-year time slice. So that's the dating on the left there that you can see. So a quick recap, really, this is the aquatic plant macrofossil record again, and you can see when you display it in this way that it really nicely captures those sort of 3 phases in the history of the plant records. So you've got this lovely diverse system, which then shifts to obviously a much less diverse system. You've lost the stonework meadows, and then ultimately into this sort of green zone there, where you're left with these very nutrient tolerant species, a really impoverished, much less diverse flora than we had in the past. So that's the plant record, and I think that's pretty unequivocal and well understood. But then if we look at some of these other remains, if you remember, Carl mentioned earlier on the Cladocera. So these are the Daphnia and the other sort of water flea species, and they leave these Aphippia, these resting eggs that we find in the sediments. And the real takeaway, without all the detail of these plots, but the real takeaway here is that we see a shift from species more associated with sort of a benthic systems, sort of attached forms, to these larger planktonic species. So we've got this planktonic dominated fauna with these larger Daphnia species. And there's also a whole host of other animal macrofossil remains, which I can't pretend that we actually fully understand what some of these are actually showing. It's quite complex, but you can see in brackets there some of the remain types. So we see caddisflies and mites, and I'm going to say a little bit more later about changes that we're seeing in mollusks. So we're seeing changes in some of the snails there, bryozoans. So these are these small aquatic invertebrates. And also in fish remains from scales and fish fragments, as well as fish egg cocoons. So some quite interesting changes there that I say we'll look at a little bit more detail in a moment. And then the diatom records, which you saw a moment ago, Carl mentioned this big increase in these planktonic species, Stephanodiscus parvis, indicative of the progressive eutrophication of the site. But there's also another interesting changes in that other species I've highlighted there in green, which is called Cochoneus placentula. And again, I'll return to that in a moment, because that's non-planktonic. And so we were kind of questioning why would that be increasing in recent times. And then the chironomids that you saw in the orange plot, where we've seen this increase in the more eutrophic and mud-dwelling taxa and a decline in the plant-associated species. So these real dynamic set of changes. And what we first of all wanted to do in this paper was to just put all that together, really, and look to see whether all these different groups were responding at the same time or not. And remarkably, the timing of these major compositional changes and all these elements of the food web are strikingly synchronous. So there really is some strong similarity in the timing, and that might not necessarily be the case. There's lots of lags and leads in different species groups. So it's really surprising to see these profound changes. So if you look at the lower one, that's dated to the early 1800s, and you can see that the plants and the Cladocera and the chironomids, they're all exhibiting this marked single sort of directional change, showing this common response to 19th century eutrophication at all levels of the food web. And then maybe even more striking, then if you look at the shift since 1900, you can see that the arrows there depicting this gradual and progressive shift, reflecting that continual turnover in the assemblages of all those different groups as eutrophication has advanced at the site during the last 100 years. So it really is an amazing and fascinating set of shifts. So what we then wanted to do was ask ourselves a few questions about what how can we explain what's going on? Okay, we know that there's this overriding eutrophication signal, but there's a lot of debate in the limnological literature about the role of nutrients, which we think of as bottom-up controls on systems versus top-down controls, which come from the food web and from grazing and from fish pressure and then zooplankton pressure and zooplankton eating the algae. So there's all these kind of theories really that we can sort of test with these multi-indicator approaches. So the first question we wanted to address was what's causing this increase in these Daphnia over the last 100 years? And if you can kind of follow the story through from sort of left to right, so in green, that is the increase clearly shown there from 1900 in the epiphyal remains. So the first thing we looked at in that first block was what was happening with the diatoms. And what it shows is you've got this marked increase in those planktonic diatoms, which is at the same sort of timing as the shifts in the Daphnia. So one of the explanations could be very much a bottom-up one. You've just got a more productive system, ample supply of food with eutrophication, and all of these algae are providing a food source for the Daphnia. So they're happy as Larry, and they've expanded in their populations. And then if we move on to the next block, which are the fish remains, we see that there's also some synchronicity there as well. There's a decline there with those arrows all shifting, sort of depicting that decline in the fish remains, indicative of reduced fish abundance really since the 1900s in the system as well. And that's also supported by the mollusk remains on the far right there. You can see that we're seeing increases in the mollusks. So these are snails essentially, again over the same time scale. So that's suggestive of a release from fish predation. The fish are not eating the snails, the fish are not eating the Daphnia. So it looks like there's a top-down control here as well. So there's some quite interesting things we can kind of unpick from that. There's certainly some evidence that there could be both top-down fish predation and bottom-up increased biomass factors that could have promoted that recent rise in the Daphnia. And then our second question was around these interesting bryozoans. So what's caused that post-1840 decline? It's a really marked decline shown in the green there with that black bar. And these freshwater bryozoans produce these overwintering things called statoblasts. And these colonies are sort of loosely attached to various surfaces in the system. So what could be driving that? And again, if you look at some of the synchronous changes, the first one there, where we know we've got these increases in Daphnia. So perhaps it's just increased competition for food. The Daphnia and the bryozoans are both competing for the same food source, perhaps. But then the other potential explanation, I think, comes from by examining this macrofossil remains from the aquatic plants. And what we know is, we've explained already, is that there's a big change in those. We've got a massive loss of habitat. So we know there's a decline in the abundance of the floating leaf plants, but also a decline in the overwintering submerged vegetation, and particularly the stoneworts that Carl talked about earlier. So there could well be a dual explanation here as well, that it could be the loss of the plant habitats, as well as changes in the food sources that appear to be, or the competition for the food sources that could be likely causes of the observed decline in this particular bryozoan. And then finally, we were interested in this diatom I mentioned earlier. This is one of these non-planktonic diatoms, what we call an epiphytic diatom. So this is typically associated with plant surfaces, and it's what we call an adpressed form. So this is Cochoneus placentula. It's very common in shallow lakes attached to plants. You can see the little photograph there. It's a sort of saddle-shaped diatom that is very flat and adheres really tightly onto the surfaces of plants, the stems and surfaces of water lily leaves, for example. So what could be driving the increase in that in post-1940? So our first thought was perhaps it was eutrophication driven. Maybe there were more plants around for this species to attach to, more plant biomass, more habitat. But of course, that isn't supported by the aquatic plant macrofossil data or the historical botanical records at all. We know that actually those habitats were in decline at that time. So then we started sort of exploring sort of other explanations, and that's where perhaps the mollusks perhaps have the answer. And here we're thinking it could well just be that because you've got more snails, the Cochoneus actually can outcompete some of the other diatoms because it attaches so closely to the plant substrates. It's pretty resistant to grazing pressure. A lot of other diatoms are sort of stalked forms, sort of upright, and they're much easier to munch away at and to, more edible forms, if you like. So it could just be that it had this sort of competitive advantage by being flat and closely adhered to the plants. And that's sort of mirrored by that little pulse that you get earlier in the record as well. So we postulate here, I think a plausible explanation is that it's resistance to this increased grazing pressure in the system from the invertebrates that could be the driver for the increase in that species. So let's try and sort of wrap all that up. I appreciate we've covered a lot of ground in the last 45, 50 minutes or so. But I think one of the really key takeaways, and Carl mentioned earlier, is that Groby Pool is fascinating, and it has to be one of the best studied shallow lakes in terms of palaeoecological and sort of historical records and our understanding of how eutrophication impacts upon a shallow lake system. And we hope we've shown you in this sort of short space of time how you can bring together all the evidence. It's sort of like all the pieces of the jigsaw, if you like. So bringing together the plant and animal macrofossil data from several cores to really reveal this sort of strong sequence of events and really very coherent patterns across all these different organism groups. And what I think essentially it shows for Groby Pool is this major shift from all these attached forms, these benthic non-planktonic forms, to this much stronger pelagic sort of plankton-dominated system over the last few hundred years, which is clearly indicative of the eutrophication, long-term eutrophication. We've also shown, I hope, that all the different components of the food web display what appears to be a really closely tied shifts in composition in response to that increase in nutrient loads. And then I think importantly, and maybe where Groby really adds to our understanding, it's not just the compositional changes, the structural changes, but really now trying to explore the causes of those changes across all these groups. And it's really helping us to understand the mechanisms and some of the interlinkages between these groups and coming up hopefully with some fairly plausible explanations as to what the range of forcing factors for driving those observations might be. And as I say, it really is like, building the jigsaw puzzle and building this more complete picture so we can really try to get a handle on ecosystem structure and function, and that can be painted by employing this full record. It was very traditionally palaeoecology was conducted as sort of single remains. The diatomists did their thing and the chironomists did their thing and the macrofossil experts did theirs. But the beauty of this, of course, is just bringing all of that together, and you can achieve so much more by doing that. But then what does it all mean, I think, in terms of the future of Groby? And this is where it'd be, fascinating to hear a little bit more from the audience. And as Carl said, we haven't revisited Groby for quite some time now. But clearly, the findings suggest that it's a heavily degraded site, sadly. It's lost a lot of its botanical interest and its biodiversity. And it clearly needs focused management and, we would argue restoration at the catchment scale. And we've been particularly interested recently in trying to understand where there are refugia in catchments and in landscapes for trying to restore lost aquatic plants. And as Carl showed you, Groby Pool is sadly sat in a somewhat of a desert when it comes to many of the plants that were formerly there. So the suggestion really, I guess, is that it may need translocation if we want to try to bring some of those lost species back to Groby Pool. So I hope we've let you into the secrets, and the secrets are no more. But we just obviously like to thank you hugely for your attention. We should obviously acknowledge the many people that have contributed to the research on Groby Pool. It's certainly not just the work of Carl and I and all the funding bodies. Much of this work was funded by the Natural Environment Research Council over the years. And then obviously we'd love to welcome any questions, and particularly to hear about your Groby Pool stories, because I say we sort of feel like we've slightly lost track of the recent picture. So thank you so much for your attention, and I hope that was of interest to you all.
Well, thank you so much, Professors Bennion and Sayer. That was an absolutely amazing talk. And who knew that the mud of Groby Pool had so many secrets inside it? I was particularly blown away to know that there's bryozoans there. Right? That's something I always think of as encrusted in a rock back in 150 million years ago. But there are bryozoans in Groby Pool. Absolutely fantastic. A reminder to everyone who's watching, keep your questions coming in. You can put them in the comments or on the chat, depending which platform you are watching on. And in a moment, our SMS text number will pop up on screen, and you can also send your questions in that way. But to start with, a quick a recap question that goes over some of the methodology that you asked at the start, and it's from Dr. Rykow, who's joining us from the University of Leicester. How do your ages established from the cause, which I'm going to add a bit to? You talked about how some of your dates from the from the 1900s were established with nuclear fallout and some of the isotopes. When you were dating lower down, is that when you were using the lead isotopes and the cesium isotopes? Is that how you, because you had a few tie-in points from the 1800s?
Do you want me to come in on that, Carl?
Yeah, go on. How do you go
so
first?
Yes, we've got slightly different approaches for different cores that we've taken over the years, but the vast majority of the cores from Groby Pool have been dated using radiometric techniques that I did mention, albeit very briefly. So yeah, mostly lead-210. So that allows us to get back to about 1800, 1850, and then we do have to extrapolate below that. So there's a little caveat obviously attached to that. That comes with a slightly bigger error. But knowing what the accumulation rates were for the kind of 1800s, 1850s, we can kind of extrapolate back to work out roughly the ages beyond that. And yes, some of the cores were also constrained by the carbonaceous particle method that I mentioned. So we also had some tie-in with those. And they're also quite clear changes in organic matter, so we can actually correlate cores. If you've got a few dated cores and then there's other similarities in other parts of the stratigraphy, we can kind of correlate undated cores with the dated ones. So I think the short answer is different approaches, but largely, by and large, it's using the lead-210 and the cesium records.
Excellent. And our local councillor representative, the councillor for Groby Pool, is watching today, Councillor Martin Cartwright. He's also chair of the Charnwood Forest Regional Park Board. And the first of his questions was, where's the nearest other site where you've got cores that you can compare to? It's I suppose it gives us, I mean, many of us watching today will have been blown away by the significance of Groby Pool, and I suppose understanding where the nearest comparable site is will help us understand a bit more about just how special it is.
I can have a go at that. I mean, I would say it's spectacularly its historical flora is spectacularly special. So there would have been sites that had a similar sort of thing in the Norfolk and Suffolk Broads, which was would have been the Mecca in Eastern England. Some of the sites might have been as good as Groby Pool in the past, I suspect. You might have had as a really diverse flora. Maybe some places in the Shropshire, Cheshire Meres, but I don't think they would have been as good as Groby Pool. I think in the Midlands region, you'd struggle to find anywhere that had that incredible botanical diversity, and people realised that, and that's why they went there, because they couldn't find a lot of these plants anywhere else around. So I think pretty special place. I don't think you could find sediment records that would give you such a diversity of species anywhere very close, because there was nowhere quite as good.
And then looking to the future, a few people have asked, is it a place where research is continuing? Are there, is there a plan? Should there be further sampling of the site? Is this a place where one could study the impact of climate change?
Do you want to go first, Helen?
Yeah, I think it's certainly from a paleo point of view, it would be very difficult, I think, to try to extract a climate change signal from a site like Groby. We've tried to do that in other shallow lowland lakes, and I think because the land use pressures and the eutrophication signal is such a sort of sledgehammer, really, very subtle climate change is then quite hard to disentangle from that. So much of our sort of climate change work tends to take place at sites where there's more sensitivity to climate. So, I mean, that's not, that's not to say it wouldn't be a fruitful avenue in some regard, but it is quite a tricky thing to do from the paleo record, where you've got a very strong other major pressure. We were actually talking about this, weren't we, Carl, but as well about going back to Groby Pool, because that's one of the things we haven't got any concrete plans to do so, and we haven't got any funding currently to do any more work at Groby Pool. But again, that's another good thing you can use paleo for, is often go back in 20 years and take another core and then look to see what that adds to the story over and above what you gathered from your core back in the year, because I think it's the year 2000 was the last core we collected. So that's 20 years or more old now. So it would be great to sort of track to see whether has there been subsequent deterioration, is there any sign of recovery. But I don't know the answer to that. I don't know, Carl, whether you wanted to say anything about. I know there's some work going on at University of Leicester, isn't there? Just trying to sort of more broadly around bringing historical records together. But I don't know the details of that too much.
Yeah, no, I think it would be, we desperately need some modern day botanical surveys, I think, to see what the lake is like now. And I think the other thing is, there's other technique to develop since we finished it. So things like ancient DNA would also be really interesting to look at for a place like Groby Pool, because you've got so many species known for it, historically recorded and in the paleo records. So it'd be fascinating to do that at the site. So actually, in a way, you asking us to do this talk sort of really it certainly makes me want to go back and do some work. So I don't think we're quite finished. If anyone's got any funding, we're there in a second.
There you go, sneaky funders hiding and watching this talk.
Yeah.
Top priority work as we move towards UNESCO designation. It needs doing. It's not just the rocks that need studying. Loads of questions coming in on the future. But before we get to the future, let's stay in the past. A question from a local resident and geology student, Bethan, who asks a very geological question. Are you worried about the effects of bioturbation? So just for everyone at home, this is the process where the animals that are living in the sediment are churning it up. And obviously, if you've got nice layers with records in, there's a risk that bioturbation is mixing up all your wonderfully preserved and in time layers. So do you have to worry about bioturbation, or is one of the glories of Groby Pool that it's not very bioturbated?
Shall I have a go? Well, I think the key thing really is that yes, there's mixing a little bit. There's always a little bit, but the accumulation rate of sediment is so fast, and the biological change is so what's the word I'm looking for? Just so rapid and profound that it's a minor process. In a way, it doesn't. It's going to be going on, but it's not getting rid of the signal. The signal is very clear. It's sort of sequential. Everything matches up. Even quite heavy things like heavy macrofossils that they just don't seem to lose their position in the sediment record. We've seen this from many other sites. So it's definitely going on, but it doesn't mess it up for us, is the answer to that, I think.
There you go. Unlike in the Swidland slate from the Cambrian period, you don't need to worry about bioturbation in Groby Pool. And we're getting lots of messages of thank yous as well. The local naturalist club send their thanks. So thank you to both of them, and thank you for tuning in. Another question looking back to the past. Susan from Northamptonshire asks about those post 1880 floral changes that you showed in your various figures, and wondered if there might perhaps be a climate control. Could that be associated with the Little Ice Age or something like that? We did wonder a little bit on that. I think eutrophication is the profound driver of the plants. It's so strong that I don't think anything
can detract from that. So I think Helen would agree, it overrides, so. But there are a few other little bits and pieces in the sediment record that I remember, did make us think a little bit about a subtle climate impact, being seen, if you know what I mean. It's things like there was some dissolution in the diatoms. There was I think there's a change in the accumulation rate. There was a sparser number of fossils a bit further down the core. So there was at one point, I think, that we thought there might be a hint of something, but it's not strong, you see, because the human impact signal is so strong and it overrides. So that's my answer to that.
Excellent. And just to say, Lizzie Harper says it's been a fascinating talk and she loves the use of cores, plus, combining those data sources together. So thank you, Lizzie, for tuning in as well as everyone else. I'm going to come to those questions about the future now. And there's been a bit of a theme. We've had Martin was asking a question about this, as was Susan as well. And we've got Jane and Margaret and some of the people on YouTube asking very, very similar questions. So I'm going to roll them all together. And it's, could the eutrophication be reversed? Do you think there is a management option for that? And then rolled into that question, do you think everything would kind of go back to how it was if we just turned off the eutrophication? Or you've already mentioned translocation. Someone asked, would there be seeds in the sediment that would suddenly, return to their beautiful 1700 selves?
Do you want to have a go at that, Helen? I'll follow up if I've got any extra thoughts.
They're great questions, and they're actually the subject of a kind of big research grant we've got at the moment, which is looking at how lakes recover, and do lakes recover, and how long does it take them to recover. So they're all very pertinent questions, and I don't think we have the answers to them all yet. Actually, I mean, I think the key management sort of challenge, obviously, is just to try to get the external nutrient loading down. And I think once you can tackle that, most of the research would suggest that for shallow lakes, it can take a couple of decades to ultimately kind of get the lake back into some sort of equilibrium, particularly if you've got this internal loading problem that I mentioned early on. So, even if you tackle the nutrients in the catchment, you might still have to wait that little bit longer before some of that reservoir of phosphorus in the sediments kind of sort of washes itself out almost, and the lake gets back into some equilibrium. So there's a lot of patience involved in shallow lake recovery, but there are some positive examples of where it is starting to pay dividends, and we're seeing algal biomass come down and balanced fish communities again, and changes across the whole sort of food web that are obviously positive signs. But maybe I'll hand over to Carl to address the bit about the things coming back, what returns, and the power of the seed bank, because that's another kind of big area of fascinating research that we've been interested in, is the whole sort of restoration ecology side of things.
Yeah, I spent my whole life working out how you restore things, and I'll give you one really clear message on that. I think if we were to improve the conditions and get nutrient levels down enough, over time, it would be amazing how you'd be amazed by how well it would recover, and it would be mystical almost. We've seen other ponds, especially with ponds and things, where you can do restoration a bit more quickly. Incredible rare species return from nowhere, either from long-term viable seeds or dispersal being much more powerful than you thought. So I think you'd have to sort of remove the nutrients that sewage treatment works using stripping to get into the catchment and change land use majorly. If you could do that and get nutrient levels down, it would recover. The challenge is getting nutrient levels down, and it would be really interesting to see what would happen. I mean, come on, let's do it. You've got some influence here. This is one of the most important lakes in Britain. Let's get in that catchment and make some changes. You might have to bring some plants in, But I think you'd be amazed by how well that would recover if you could get nutrient levels down.
Bring back the stoneworts.
They come back. They're waiting.
Well, there you go, ladies and gentlemen at home. Thank you so much for joining us today for this very special first, hopefully, of many live online talks about the various aspects of heritage and the natural world within Charnwood Forest. I knew we were in for an interesting talk today, but I think the audience will agree with me. We didn't know how interesting, and we were perhaps oblivious to just how significant Groby Pool is. And so thank you so much to both of our fantastic speakers tonight on behalf of the Geopark, the residents of Charnwood Forest, and all of the Charnwood Forest team. Thank you so much. You've really brought it alive tonight and given us a call to action. And hopefully this isn't the last we'll be seeing you. Maybe we can tease you back to Charnwood Forest for some more coring. I'm already having messages saying we must core more. So Charnwood Forest has now got the bug for coring of Groby Pool. So there we go. And just a final thing to all our viewers at home. If you'd like to stay in touch with everything that's going on in Charnwood Forest, where to go and see these wonderful things, there is more to Charnwood Forest than just the sites that you've probably heard of. There are many hidden treasures, and you can find out about some of those on our website, charnwoodforest.org. You can also learn a lot about our heritage and our history. You find out about the projects that we're working on, funded by the National Lottery Heritage Fund, all the partners that we work with, and you'll be able to keep in touch about future events like this by signing up to our newsletter. You can find all of that and so much more at charnwoodforest.org. But at that point, I will thank our speakers once again. Our viewers at home, thank you so much. And we'll see you next time for a Charnwood Forest Geopark live talk. Thank you.